All right, so now we are going to take a look at the different types of cell receptors. So again, talking about a hydrophobic signal that would need to bind to a receptor in the membrane as opposed to just diffusing through the membrane and dealing with an intracellular receptor. So there's basically three types of cell surface receptors. The first is an ion-coupled receptor. The good news is we've already learned about this one. These are our ligand-gated channels. This is what we talked about with neurotransmitters. They are able to take a chemical signal in the form of a neurotransmitter and transduce that signal into an electrical signal by opening their ion channels, which changes membrane permeability. So we've already learned about one of them. Yay! We haven't learned about our G protein coupled receptors or our enzyme coupled receptors though. So that's going to be the focus of the rest of our chapter here. So we're going to start with the G protein coupled receptors or the GPCRs. Okay. Then a little bit later we will move on to our enzyme coupled receptors. So just to, give you an idea of the importance of these receptors, here's a few different things that are going to utilize these different receptors. So something like nicotine or your benzodiazepines, things like Valium, Ambien, things like that. Morphine is going to actually work on a G protein coupled receptor, which also means that heroin also does this. So, we're kind of in the middle a little bit of a heroin sort of resurgence in the epidemic here. And this is a G protein coupled receptor issue. Capsaicins, so if you like to eat spicy food, that's an ion channel. Okay. So, again, just to give you the idea of, the overall daily importance of these different types of receptors. All right, so let's start with our G protein coupled receptors here. This is just a generic structure here, so this is not a specific one, but what we see is that we have a multi-pass membrane-bound receptor. And each of these will consist of three different domains. We're going to have the intracellular domain, okay? We're going to have the extracellular domain, and then we're going to have the transmembrane areas. These receptors have 7 different transmembrane domains, so it's going to weave its way through the membrane 7 different times. Okay, so hopefully looking at our N terminus here and our C terminus here, you're starting to kind of think about, oh, hey, what would the signal sequence be that would create this structure here? So when we look at the overall structure, we can see that there's going to be a signal binding site. So this is where our extracellular signal would bind. And then in the intracellular domain, we have a portion that's going to interact with what's called the G protein. There's a lot of different types of G proteins and even more types of G protein coupled receptors. In humans, we've identified over 700 different G protein coupled receptors. And if you can believe this, because, we study mice a lot, they have over 1,000 different GPCRs involved in their sense of smell alone. Okay, so these are pretty significant cell receptors. Because of that, and because they're involved in so many things, they are very active as drug target sites. So if we can activate or deactivate these receptors, we can have pretty significant physiological changes. All right, so let's take a look at what the G protein is that these receptors are interacting with. So here's our receptor, our transmembrane receptor weaving through. So it's got its extracellular domains, its intracellular domains, and then its transmembrane domains. They are going to interact with what are called G proteins. So G proteins are trimeric, meaning that there are 3 different subunits to the protein. So here you're seeing an inactive G protein. It's inactive because it is bound to GDP. So remember, we talked about that before. So if it's bound to GDP, it's going to be in its inactive state. At this point, in an inactive state, you could be thinking, what would activate this? GEF. Okay, it would cause it to drop the GDP, grab GTP. Okay, so just kind of building on what we've already talked about here. Okay, so we have 3 subunits. The alpha is the biggest, okay, and it's what actually binds GDP. Beta is in the middle, and gamma is farthest away. So you got alpha, beta, and gamma subunits. Now, alpha and gamma are lipid linked to the membrane, which means that they are completely anchored in the membrane, and beta gamma always stay together. Alpha can disassociate from this, but beta gamma are always going to form the beta gamma complex. Okay, so when we bind our extracellular signal, this G protein, okay, this entire G protein will laterally diffuse, okay, and we'll see the interaction of this domain with the G protein. This is going to cause GDP to dissociate, which is going to free up the binding site for GTP. Once we have bound GTP, we're going to split into the alpha and then the beta gamma complex. These are both now in active states, which means that we can have 2 intracellular pathways here because we have 2 activated effectors here. So we are able to actually, utilizing one cellular molecule or signaling molecule, we're able to actually activate 2 pathways inside of the cell. Now, how do we shut it off? In most cases, constant signaling is not always going to be helpful. So we also have to be able to shut this entire thing off. So the alpha is a GTPase, which means it has the ability to hydrolyze GTP to GDP. So we've talked before about how we're going to have an inactive protein when we're bound to GDP. So what happens is we're going to hydrolyze our GTP, forming GDP, which shuts off the alpha complex. The inactive alpha complex is then going to reassociate with the beta gamma complex, which shuts that off. Okay, so to shut it off, we need to hydrolyze our GTP, which inactivates alpha. Alpha then reassociates with the beta gamma complex, and that reassociation is going to shut it off, and we're back to a complete inactive G protein. Okay, so that's the basis for how do we turn it on. We're going to need GTP. How do we turn it off? We're going to hydrolyze GTP. Now, there's a handful of bacterial toxins that can actually affect these G proteins. 2 of them are the toxins that are going to cause cholera and pertussis, which is more commonly known as whooping cough. You guys are going to be looking at these two diseases for your first assignment and figuring out how they are affecting these G proteins and how we end up with the overall effects, the signs and symptoms of these different diseases.